Hey bio students, welcome to today's lesson. By now you should have finished the bell work on dihybrid cross practice, so take a look at it and let's check it. Make sure that you pause and make any corrections if you need to as we go through. So the bell work says that tall plants, big T, are dominant over short plants, little t, and purple flowers, big F, are dominant over white flowers, little f. What is the chance that a plant that is heterozygous for height and homozygous recessive for flower color, and another plant that is also heterozygous for height and heterozygous for flower color, would have offspring that is short with purple flowers? So remember, we are looking at two different traits at the same time. So our first parent is heterozygous for height. So we know that they have to be big T, little t. That same parent is also homozygous recessive for flower color. That means they're little f, little f. So there is our first genotype of the first parent. The other parent is heterozygous for both traits. So they are big T, little t for height, and big F, little f for flower color. So there is our first genotype cross. The next thing we want to do is separate and look at each trait individually. So we'll take a look at the height trait over here and the flower color trait over here. And after you fill in the Punnett square, you can see that there's a 3/4 chance that the offspring will be tall, a 1/4 chance that the offspring will be short. And over here, there is a 1/2 chance for purple flowers and a 1/2 chance for white flowers. But the question's asking, what are the chances of the combination of these particular traits? So what we need to do is figure out the probability for these different combinations. In order to do that, all we need to do is multiply our fractions together. So we know that there's a 3/4 chance of tall and a 1/2 chance of purple. So we multiply 3/4 times 1/2. Remember, you just multiply across the top, multiply across the bottom, and we get 3/8. Same thing with short and purple flowers. There was a 1/4 chance of short, a 1/2 chance of purple based on our previous Punnett squares. Multiply across the top, multiply across the bottom, we get a 1/8 chance for short and purple. You'll do the same for the tall with white and the short with white flowers. If you need to, pause or go back, make changes or corrections on your bell work so you know exactly what to do. All right, today what we want to do is to continue to predict outcomes of genetic crosses by recognizing variation in traits, interpreting how genes affect physical traits, and using Punnett squares to predict the traits of those offspring. So let's do a little recap about how we can interpret how genes affect physical traits and how we can use Punnett squares. So if you could, take a look at the back side of your bell work. It says Intro to Non-Mendelian Genetics. Pause the video here and complete this handout that's on the back of your bell work. You'll notice that the first cross is something that you're very familiar with. The second two crosses are new scenarios that I'd like you to see if you can try to figure out. We'll learn about them in just a little bit. So pause the video here. When you're done completing this handout, then start the video back up. All right, by now you're done completing that Intro to Mendelian Genetics, Non-Mendelian Genetics handout. Let's do a quick recap on regular Mendelian genetics, what we've known so far. So remember, Mendel studied pea plants, and he was able to figure out a lot of stuff based on those studies. So remember, he took purple flowers and he crossed them with white flowers, and he got a first generation with 100% purple flowers. That's because the purple was dominant over the white. When he self-pollinated the purple flowers, he was able to see that in the second generation, that white trait came back, that recessive trait came back. So from these observations, he was able to discover that most traits are controlled by a single gene, and there's only two alleles, two different versions of that gene. For flower color, for example, there's purple or white. And one of these alleles is completely dominant over the other. So the purple was completely dominant over the white, which is why we lost that white flower color in that first generation. So let's check our Intro to Non-Mendelian Genetics number 1. Here in this scenario, we've got big E. Big ears are dominant over small ears. We're crossing two heterozygous mice. So your Punnett square should look like this. Your genotype and phenotype ratios should look like this if we're following Mendel's rules. So again, pause if you need to make corrections on your Intro to Non-Mendelian Genetics cross number 1 if necessary. If not, let's keep moving on. So let's take a look at this scenario. We've got a red flower crossed with a white flower. What would Mendel do? Well, Mendel knows that the red flower is probably dominant over the white flower, so he would predict that 100% of that first generation would be red flowers because the red trait covers up, is completely dominant over the white trait. And then he would predict that that second generation would have 75% red and only 25% white because that white trait comes back as a recessive trait in that second cross. Let's take a look at what really happens with these particular types of flowers. When we cross a red flower with a white flower, we actually get 100% pink flowers. This is a little weird if we're following Mendel's rules, because we would assume that those flowers should be red. If we were to take and self-pollinate these pink flowers, we would notice that 25% of the second generation would be red, 50% would be pink, and 25% would be white. Again, this doesn't really match with what we've learned so far. That's because when we look at interpreting how genes affect physical traits, they can be affected differently depending on how those genes interact. So we're going to learn today about 3 different types of gene interactions that do not follow Mendel's rules. And in fact, most of the traits that we talk about or that we're familiar with, eye color, hair color, height in humans, follow rules that are more similar to these that we'll learn today than the ones that Mendel outlined as dominant or recessive. So take a look at the notes handout. It says non-Mendelian notes, and we're going to take a look at 3 different types of non-Mendelian inheritance. The first is called incomplete dominance. This is when one gene, one allele, is incompletely dominant over the other. What that means is that when we get a heterozygous genotype, we have an intermediate blended phenotype. For example, this red flower here has 2 dominant alleles, big R, big R. This white flower here has 2 recessive alleles, little r, little r. This pink flower has 1 of each. And because the red is not completely dominant over the white, some of that white trait shows through with the red, and we get a pink intermediate. So let's take a look at your notes and we'll do a practice problem. Let's cross 2 pink flowers. So they would be big F, little f, and we'll fill in the Punnett square. Go ahead and fill in your Punnett square, and you will notice that we have a kind of a normal Punnett square here, but the trick comes in with interpreting what these genes mean. Remember, 2 big F's mean red, 2 little f's mean white, a big and a little don't mean red anymore. With incomplete dominance, the traits blend and we get pink. So let's take a look at our genotype probabilities. Okay, we've got 25% big F, 50% big F, little f, and 25% little f, little f. Normally we would assume that these 3 out of 4 would be red and this 1 out of 4 would be white, but remember, this is not a completely dominant recessive type of trait. This is incomplete dominance. So we still have our 25% white flowers, we have 25% red flowers because they have 2 red genes, and 50% pink flowers because they have 1 red gene and 1 white gene, and those genes blend to bring about our blended trait. Pause the video here if you need to finish taking notes on Mendel number 1. If you're ready to go, let's keep moving on. Let's check that intro sheet that we did to see how you did. That was the 3rd cross on your intro sheet on the back side of your bell work. We had red flowers are incompletely dominant over white. We're going to cross a red with a white flower, and so you should see that we should get all big R, little r offspring. And because red does not completely cover up white, all of the offspring in the cross would end up being pink because the traits blend together. Pause the video here and make corrections to non-Mendelian genetics intro number 3 if you need to. All right, now grab the practice handout that you picked up when you walked in, and we're going to pause the video here, and you are going to take a few minutes to complete part 1 and part 2. Just part 1, numbers 1 and 2 of the practice handout. These are 2 different crosses. In the first one, you're crossing radishes, use the letter R. In the second one, you're crossing rats, use the letter Y. When you are done with the practice, all of part 1, go ahead and play the video so you can check your work. All right, you have completed part number 1 of your bell work, and let's go ahead and check it. So in radishes, we've got a gene that is incompletely dominant over another gene. We're going to cross a purple radish with a white radish. A purple radish is the intermediate, so that is our heterozygous genotype. A white radish is recessive, so we've got little r, little r. So we're going to fill in our Punnett square, like so. We have 50% of our offspring with big R, little r. We have 50% of our offspring with little r, little r. Big R, little r is purple, so we would have 50% purple. And 50% of those are little r, and those would be white. So our offspring would be 50% white. Our next one, we're going to cross 2 cream rats. Cream rats are the intermediate heterozygous trait. So we are going to fill in our Punnett square like so. And then we will do our genotype probability. So we've got 25% big Y, 50% big Y, little y, and 25% little y, little y. That correlates to yellow rats, 25%, 50% cream, and 25% white. Pause here if you need to make corrections, and let's head back to the notes. Take a look at this flower. Again, this flower is doing some weird things. We don't see completely red, we don't see completely white, and in this case we don't even see a blended pink. We see red and white at the same time. This is an example of codominance. When we have codominance, back on your notes, make sure that you're filling this in, the heterozygous genotype distinctly shows both phenotypes. We see distinct red, we see distinct white. It's because both traits are dominant. When we are using this type of dominance, we want to make sure that we're using 2 different capital letters. Let's take a quick practice on your notes. So let's say I have a red fish and a blue fish. Red fishes are big R, blue ones are big B, little b. So when I cross the 2, all of the offspring are going to be heterozygous. They'll have one red trait and one blue trait. And instead of red covering up blue or blue covering up red, or instead of the traits blending to make purple, these fish are going to come out red and blue spotted. We'll see both traits. Go ahead and finish jotting that down on your notes. If you need to pause the video, go ahead and do so. Don't forget, you want to write your genotype and phenotype probabilities. Let's take a look at a quick human example. A great human example is sickle-cell anemia. Normally, your red blood cells are nice and round, and they carry oxygen really well, and they pass through your arteries and veins really easily. People who have normal, healthy blood cells have the genotype big N, big N. Okay, these genes code for healthy red blood cells. But some people have sickle cell anemia, where their blood cells are this shape. And this is a problem because they don't carry oxygen quite as well, and they can get jammed up in the small capillaries in your bloodstream, and they can cause blood clots. People with sickle cell anemia have this genotype, big S, big S, and so they have this sickle cell trait. Well, what happens then if we take a person with normal cells, big N, and mate them with a person with sickle cells, big S, little s? Well, all of the offspring will have the genotype big N, big S. They'll have one gene for normal and one gene for sickle cells. What will they actually look like? Well, half of their cells will be normal and the other half will be sickle-shaped. So this is an example of codominance. The normal cells and the sickle cells gene are exactly equally dominant, so we see equal traits in the end. Let's take a look at your intro to non-Mendelian genetics number 2. You have a black chicken crossed with a white chicken, and so we're going to get offspring with one black gene and one white gene. All of those chickens will be black and white, or what we call checkered. All right, this is the end of the first part of our video today. When you go ahead and pause the video here and do part 2, which is our codominance. When you are done, play the next video in the series to check your work and learn about sex-linked traits.